So my name's Jeroen. I'm one of the bioinformatics coordinators of Terra Oceans. I have a lab in Leuven in Belgium. It's only an hour's train ride away. And I'm here to talk to you about the projects that has been dubbed as the Oceans Facebook. We're sort of been looking at all of the interactions between the organisms among themselves and the environmental conditions. So this is the first of two talks trying to integrate all the data sets that you have seen earlier this morning. So this project started off with us asking a question. It was actually a very simple question, like how much of the community structure, so the composition of the organisms, how many there are of each kind, across all of the kingdoms that we're looking at, the viruses, the bacteria, the protists, can be explained just by the environmental conditions. Yeah. And so there's several techniques to analyze these things, and we used a few. But whatever technique we used, the answer was always, well, not that much, right? There is an effect of environmental conditions, and especially the bacteria, as you've seen with the talk from Shini, are affected. But on the whole, we only could find a modest contribution of the environment on the community composition. And so basically with a technique called variation partitioning, we could find 18, let's say 31% if we take geography into account. With models built on random forests, the large majority of the models that we tried to build that used environmental conditions were actually not better at predicting the community structure. So it seemed that somehow, if you look at it from the data perspective, the community composition was not that well explained by just the environmental conditions they lived in. So there is something else, right? And that something else, we think, of course, is the biotic interactions, is the interactions between the species, that these interactions also govern the composition of the world's oceans. And so these interactions, they can come from very different, in different types of flavors. You can have parasitism or predation, you can have competition, you can have symbiotic relationships, and they all vary in the degree of investment of one of the two partners in the relationship between these different organisms. And of course, you cannot observe all of these interactions between these organisms as you would in a field, right? People from macroecology, they go to the savannah, they take their binoculars, and they look at the interactions between the different organisms. They look, do they compete? Do they interact? Do they breed? Etc., etc. Do they eat each other? And so that is something we cannot do at a large scale in these oceans. So we have to use tricks. And in this particular case, we're using a trick which is actually very old. It's a trick invented by Jared Diamond. You might know from the books on the history of mankind. And he came up with a technique which is based on checkerboard patterns. So what he did was he went to islands, and in those islands he looked at the birds that were present there. And he looked at when do I see one bird there in relationship to another bird. And so when he saw two birds always occurring in the same place in the same time, he hypothesized that these birds had some kind of positive interaction. There was some kind of reason why they were always together. And this reason can be many things. The reason can be that they actually have a symbiotic relationship, that the one does something for the other, etc., etc. It could also be that they just occupy a similar niche. At the same time, if two species never occur together, this could be an indication of a negative interaction or a competition. You see the one, and then you never see the other. You see the other, then you never see the one. And so it's these kind of patterns that you can use to start hypothesizing, inferring species interactions from observations of organisms. And so this is mainly a statistical bioinformatics exercise from the start. So you start from these kind of these big matrices where you have all of the different samples that we're looking at. So here would be the Terra stations. Here would be all the thousands and thousands of OTUs that Colomban and Shini talked to you about. And so we'll just look at patterns, and we look at whether we see significant overlap, significant similarity between these different patterns. And so you can measure that, and you can build distance matrices from that, etc. You can do then statistics, and then you check of all those similarities that we've seen, how many of them are really significant after testing, and then we can build them together in such a network. We built this network of interactions based on the principles that I've just explained to you. But of course, there's a lot of method development behind of this, because it's not so easy to do these kind of things, and you can have hundreds of different types of distance metrics that you can use, and they all capture different types of associations that you might see. You can also you can do pairwise interactions. You can look at which two species pairwise interact, but we know in nature interactions are also sometimes threefold, or a combination of species determines another species being present, etc., etc. And so there's tons of different methodologies to capture all these things. And so the approach that we use was like, given that many different tools have very different readouts, why not combine them? And so we built this, and this was work we've done before, we built this ensemble approach where we take all of the data and we use all different types of, a selected set of different types of metrics and linear models to combine and then determine this interaction network. And so this was developed, as was said before, in a completely different project where we worked on the human body, the bacteria on the human body. And I'm just showing this. The reason why I'm showing this is, but at that time we were really proud. We were like, yeah, we have this huge network of the human body, and we've done so well. And then came the Tara data. And the Tara data turned out to be a huge nightmare for these kind of approaches. And the reason for that it will become very clear very soon. Is first of all, we're working across 3 different kingdoms. We're working with environmental contextual data. We're working, again, as you've seen before, 68 stations, 2 depths, 7 size fractions, and then all our parameters and everything. But the thing is, of course, we wanted to look at all the interactions. So what you want to do is you want to compare every single size fraction with every single, so it blows up exponentially. And so basically, in the end, we after cutting and cutting and cutting, we were still stuck with 23 independent networks that we then somehow had to merge and combine. So we were looking at the network of networks. It was. I mean, Gypsy still has a headache when she thinks of that period, but. And also the computation was quite significant. It took, I mean, in the beginning, our algorithms were slow because they weren't up to this, so we had to improve. And by the end, we were getting really good. But still, it was a full month of full-time computation on a 400-node cluster. So that's 400 of these laptops working for a month just to do the basic computation, right? So this was pretty significant. And then we got the global ocean interactome, right? This is the network of networks. It's also known as the hairball. And so what you see here is basically everything piled together. There's nothing you can do with this, right? But this is the whole data. This is the global predicted interactome. It has 90,000 taxon interactions predicted. So just to give you an idea of it, to start analyzing such a thing, it is truly a challenge. And therefore, we started analyzing it, but as has been said before, we see this more as a resource. We see this more as a starting point for everybody else to now go dig in and find some gold nuggets in there that we hopefully missed. But another issue with all of this is, what do these associations truly mean? Because that's always a big issue. If 2 species co-occur, does it mean that they are truly interacting? Because co-occurrence can mean very many things. And the most obvious of these things is, for example, this concept of niche sharing. If 2 organisms, you always see them at the same time in the same place, maybe they don't like each other, maybe they just like that temperature or that pressure or that time of year, right? And those kind of things. So we have to there's this environmental component in there that we have to filter out somehow if we really want to go after the Facebook of the ocean, that the true biotic interactions between the organisms. And that's a development that also we did for this particular project, trying to filter that out. And I can't really go into the details of this, but basically what we did is we took that network that you see at the far left, and then we zoomed in and we looked for these triangles. We looked for triangles where you had 2 species interacting and there was one environmental factor associated to these species. And that triangle would tell us, there might be an environmental effect on that interaction that we see. And then we use 3 different approaches, I won't bore you with it, to figure out whether it was the environmental factor that determined that association or whether it was truly an association between these species. Long story short, we could, for the 90,000 interactions that we had there, for 11,000 we could actually show that it was environmentally driven. And that sounds like a lot, but for me it's actually very few. I would have expected that a large majority could be explained by this. Of course, there's a bit of a trade-off. Here, it's only 12% that we see driven by the environment. It could also be that our methods are still not up to scratch of detecting all environmental effects. This is the limitation at the moment. Anyway, if you have an environmental factor affecting such an interaction, what is it then? We see that is not everything. It's phosphate and nitrate as nutrients seem to be a factor that have a big effect, as well as temperature. And it's also not all organisms, right? You see that the distribution of organisms whose interactions are affected by the environmental factor are also different. It's also the different types of trifluses are different. What we see most, for example, is one environmental factor being negatively affected to 2 co-occurring organisms. Again, these are overall statistics. They're just for us to learn what kind of patterns are out there. Another big issue in all of this is it's easy to predict 90,000 associations, to have a hairball of 90,000 organisms. But how do you know whether what you're predicting is actually true? How do you know whether your method is good? And this is a very tricky business, because if you want to do this, what you need is a gold standard. What you would preferably need was a data set in which you knew all the interactions, all the ecological interactions, and from that data set, you would do your analysis and you would see how much you would predict. But that's very difficult to get in ecology, and especially in microbial ecology. And so what we did as second best, let's say, we used our gold standard list. This was compiled by Nico in Colomban's lab, of known interactions. And so it's an interaction, it's a data set of a few hundred interactions that are described in literature. And what we did was, well, look, in our data set, if we see either of these 2 organisms, do we also predict that interaction that we know in the literature? And so what we had was that we had 42% of a thing. So we have a sensitivity of 42% of predicting what is already known. I would consider that a success, because it's given the idea that it's based on principles and then a lot of filtering, that we can almost predict half of known interactions. I think that is very, very good. What is interesting is that if we only focus on parasitic interactions, we get even better prediction. And that's something I want you to keep in your head. What I think is even more important, we try to also assess the false discovery rate. So how many things do we over-predict? Right? And that turned out to be very low. And the reason for that is because we're being extremely strict in our parameters. We use extremely low p-values. We do hundreds of different types of tests to cut down the noise and to cut down, because to over-predict is you're not doing anybody a benefit here, right? You want that list that you're giving to the community to be at least as conservative as possible, right? If you might want to miss things, but you don't want to over-predict. That's my personal feeling. So basically, we sort of established the validity of our approach, and then we started looking at, okay, what are the global patterns? And so what we see is that not all species interact the same way. You clearly see patterns that different organisms have different, levels of interaction. What we see, for example, is that the Syndiales are very highly connected. So they have a lot of interactions with lots of different organisms. We see that the diatoms are mostly negatively interacting. The arthropods are also more negatively interacting, etc. And what you see also is that you have local and global patterns. So sometimes an interaction is based on a global pattern. You look across the world, and you see everywhere on the world you look, you see that one is there and the other is not there, and that one is there. And sometimes the pattern is just locally. It's driven only by Mediterranean samples, only by Indian Ocean samples. And so, and we sort of estimate that 13% of those interactions are local. What is interesting, though, is although the overall trends are the same, what you see here in the bottom are the interactions that are in different ocean basins and or regions, and that you see that the interaction patterns actually differ in different areas of the ocean. And so these are examples to just give you a little bit of a flavor of the types of patterns that we're comparing. These are examples of these local patterns where you have patterns. So here in the dashes you have one organism, and the full line is another organism. So you see this is a nice global pattern where if this one goes up, the other one also goes up, etc., right? But you also have local patterns, for example, Mediterranean sample or a South Pacific Ocean pattern, etc., just to give you an idea. And so then we started to analyze things, and we were noticing there's a lot of parasite interactions here. So that seems to be, it seems to be a recurring theme across the whole of Terra. The huge underestimated diversity of parasites, and also for us in our data, they always popped up as being much more interactive, etc., etc. And so we looked at, so okay, if we look in the global food web, if we look into the functional groups, where do those parasites affect things? And basically they affect across the globe. They seem to be interacting with both the heterotrophs and the autotrophs at the various scales. Oh, I'm almost there. And we see that they're affecting a large number of different groups. And we also see those parasites using the microscopy done at Roscoff and Embl. We also see interactions from phages, so phages to bacteria, infecting bacteria. So we've identified a whole bunch of novel phages for various bacterial groups that were never seen, as you can see here. For example, we have here a Verrucomicrobia phage, an archaeal phage that was never known, etc., etc. This one, so there's a lot of novel things that really need fleshing out, where we scratch the surface, and then it's up to everybody else to help us to dig deeper into that. The nice thing, and that's one of the last things, the nice things that also came out of this is you have predictions, but in the end, it is predictions, right? It is still the product of a bioinformatician sitting behind a computer, and so you never know, is this really real, right? And so for some cases, we went into the lab together with the people from Colomban's lab to really, and Chris's lab, to figure out, are these interactions truly true? And this is a very nice example where a predicted interaction in a computer program, if you go back to the samples, you put them under the microscope, you actually see the interaction. This is an acyl worm, which has here the red here are green algae that are living inside the worm and doing photosynthesis and giving energy and nutrition to the worm. And this thing here was predicted by just looking at a pattern like this. So I think that is very, very exciting. Okay, so as I said before, this is a resource. We have a website. If you're a bioinformatician, you can just click this button and you can download 73 megabytes of tables to crunch to your heart's delight. If you're slightly less anxious about, slightly more anxious about doing those kind of things, we've also provided this interface where you can actually go in the network, zoom into the hairball, start clicking on things and seeing like, oh, what's this then? Oh, that's what's that, and it tells you what it is, etc. So you can browse the interactions as you go. It's all on this website. If you have your own data sets, the software that we develop is also available. Go to our lab's website, go to software, and then the tool is called CONET. And with that, I have to acknowledge a great number of people, because if you integrate, you work with everybody, which is amazing and sometimes a challenge. But anyway, the two people that I really want to thank here are Gypsy and Caroline. You see their faces, they're sitting there. Those were the drivers of the project, together with Nicolas from Colomban's lab, the Chris Bowler's lab, Matt Sullivan, Per Borgs, Sylvia, all have been helping and driving this project. And a very, very, very special thank you for Eric and Steffi. You've heard her name before, you'll hear it again. They've been driving the whole of Terra, and we have to be very grateful for that. And with that, I thank you for your attention.